Welcome to Let's Talk Space Medicine. I'm Kyle Anderson, your medical student, or now doctor, space nerd host. For today's show, I'm excited to introduce Dr. Eliah Overbey, a University of Austin, Texas professor in bioastronautics. And a lot of her work is involved, Blue Origin, SpaceX, collecting samples from astronauts and doing a lot of work on their genomics, proteomics, and all the omics. And we're excited to talk to her today about her journey. So welcome on the show.
Thank you. Thank you so much for having me.
So where are we right now?
So we're at the University of Austin, not the University of Austin at Texas, but the newly founded University of Austin. This is our second year of operation. We have freshmen and sophomore students who come here to study the liberal arts. We're a liberal arts university with a strong focus on computer science, programming, and artificial intelligence.
Just a few short topics, that not topical at all in terms of recent advancements. So wow, a lot of interesting work, and tell us a little bit about your particular role.
Yeah, so I'm an assistant professor of bioastronautics. I study changes in astronaut health, specifically changes at the genomic level. So I've worked with SpaceX astronauts, with the Inspiration4 crew, also with the Polaris Dawn mission, collecting biospecimen samples, blood, urine, stool, saliva, body swabs. From Inspiration4, we got skin biopsies, so we could look at different layers of skin at the transcriptomic level of gene expression. And then we've also, working with the group at Cornell, I work very closely with Christopher Mason's lab. I did my postdoc there. So working with them, we've also collected from Axiom 2 astronauts, so this was John Schaffner and Peggy Whitson's mission a few years back, and also with Blue Origin astronauts, which is just a short 8-hour drive from here in West Texas.
Short 8 hours, straight into the desert, right? Feels like...
Keep an eye on those gas stations.
So a lot of people in my little space medicine community are interested in, as this field grows and as you're championing your efforts here, they want to know, trying to highlight people's journeys to see if they can maybe see how their particular skill set can align with this ever-changing field of space medicine. So I kind of want to hear what is your origin story? When did you figure out that space and medicine can even go together? Because I feel like when people first find that out, it kind of opens up a whole new world, and then they get sucked in, or at least that's my case. So
yeah.
I'd love to hear your origin story.
And people also seem to figure it out a bit later in their educational journey. There's not, programs right out of undergrad that are for space medicine. It's something you get to later on, either in clerkships or in grad school. Where I first saw it was, I was in graduate school. I was doing my PhD at the University of Washington in the Department of Genome Sciences. I was studying different emerging omics technologies. So I was using nanopore sequencing to look for RNA modifications. And I was using a method called, it was cutting edge at the time, but there's been, I think, improvements on it now, called Hi-C, that looked for changes in chromatin conformation. So your DNA, when it's in your cell, it's not sitting there as a two-dimensional object as we see it on pages and textbooks. It's a dynamic three-dimensional system, and these three-dimensional interactions have functional consequences. So I was doing some profiling of different cell types, looking at their different configurations. While I was doing all of that, a news article caught my attention. This was when Kate Rubins was in space, on the International Space Station, and she did DNA sequencing in space for the very first time. And that got me thinking, oh, what do we know about, because we study all these different diseases on Earth, but surely astronaut genomics are also changing. The three-dimensional shape of their genome, their gene expression, the protein profiles. I mean, the DNA itself might even be changing from radiation damage. So I was curious, how much of this have we actually studied on astronauts? And it turned out we hadn't studied it at all. This was before the twin study. So this was, not only was this the first time DNA sequencing was done in space, astronauts weren't even being studied when they came back down to Earth. And Kate Rubins' experiment, it was a technology demonstration experiment. They actually prepared the DNA libraries on Earth and then just thawed them and ran them through the nanopore sequencer in orbit. So that was really sort of the birth of any sort of genomic studies in spaceflight. So there wasn't a whole lot to get involved in at that point. I was fortunate through my searches, I found that even though humans weren't being studied on the International Space Station, mice were. C. elegans was, Drosophila was. So the biological and physical sciences program at NASA were taking studies of these organisms on the ISS and then sequencing them back on Earth and making that data public. So they had what was then called Gene Lab, what is now called the Open Science Data Repository. They have these analysis working groups. So I joined their animal AWG, and I just started getting to work analyzing data. My first publication was out of that AWG.
Mm-hmm. Shout out to NASA Ames and all the great folks out there that are part of that biorepository. It's an incredible resource to check out, and yeah, I mean, what an introduction. So you were in grad school, you see this news article, then you got involved from there, and just everything kind of unraveled. And
then I very closely followed the twin study. Mm-hmm. When that came out, I paid attention to one of the people doing a lot of the different omics work on that samples. There were different teams studying different things. Scott Kelly was studying across a variety of different modalities, omics being a part of that. The person who was doing the omics studies is a scientist called Christopher Mason. He does work out of Weill Cornell Medicine in New York City. I reached out to him, I believe it was, I met him at an ASGSR conference. I found out at that same conference from my NASA Ames internship mentor, John Galaska, that there was a postdoc fellowship that NASA had opened up that I could apply for. So I found that out at the conference, and I saw Chris Mason there. Mm-hmm. And so I went to Chris Mason and was like, I'm going to apply for this, and I'm going to come to your lab and work more on astronautics. And he was like, great, yes. Go do it, go write it. So I wrote that, I sent it to Cornell, to Chris, and he connected me with Cornell's grants department. I finished the grant with the grants department. I didn't talk to Chris for like 8 months in that interim period, because we were just waiting for the grant to arrive, but also COVID had happened. Mm-hmm. So everyone was kind of doing their own thing during that time, figuring out sort of a new working landscape. Nobody could really go into the office. Things have changed quite significantly. But then I found out 8 months later that I got this fellowship. So I called him up, I emailed him up, and I was just like, hey, guess what, I'm coming.
Remember me? Yeah.
Yeah. So I showed up on his doorstep, or on his lab doorstep, with a NASA fellowship, and it's hard, it's easy to get hired when
you bring your money
with
with
you.
you.
Money, yeah.
Yep.
So that started that journey. Day one, when I joined his lab, he was like, all right, sit down, we have a meeting with SpaceX. My NASA fellowship wasn't for any sort of human work, actually. It was to look further into changes in gene expression in retinal tissue in mice. Can't really get retinal tissue from humans to study, so.
What? The IRB wouldn't let you do that?
Yeah, nobody wants to give up their retinal tissue yet, so, I don't know, but. So I came into the lab expecting to be working mostly on mice, maybe getting access to this twin study data that had now been published, but was very difficult to actually get your hands on. You have to kind of be in one of the labs working on it to have access to that data. But day one, he sat me down on a meeting with SpaceX, and I found out about this mission called Inspiration 4.
Yeah, I've heard of that one.
Yeah.
What an incredible opportunity. I mean, like, talk about hitting the striking while the iron's hot. I mean, or being there in the right place, the right time. That's an incredible time to be part of the first private space mission to go to orbit. That's just, I mean, inspiration. So
exactly. Yeah. So that was the first time I heard about the mission. We wanted to do something, an upgraded version of the NASA twin study for the Inspiration 4 crew. And the things we wanted to upgrade, we wanted to use newer omics technologies. We wanted to do a wider variety of different types of sequencing on them, so expand the types of data we were collecting. And we also really wanted to get it into researchers' hands. Since the NASA twin study data was so hard to distribute, working with a commercial company was an opportunity to actually create a scientific study structure that could be disseminated to a wider research community.
Yeah, it really feels like the big challenge these days is the transition to getting data in the hands of researchers. I know that like a lot of the folks at Trish, the Translational Research Institute for Space Health at Baylor College of Medicine in Houston, they're trying to put up this whole architecture for how commercial space flight is going to relate to how they get the data out, and they're trying to build this whole framework, and it's a big heavy lift, so I'm sure that was a big challenge.
Yeah, so we managed to get through that process. We have that data for Inspiration 4. What I was doing there, I was flying around to all of these different sample collection sites across the country. I was in Hawthorne, at SpaceX's headquarters when the crew was there to get samples from them. I was in Florida with them. We did some sample collection in Houston. There was one time point we wanted to sample them at where they weren't all in the same place. So we had to create these custom kits that we sent to them in the mail, and then we also worked with a company called Exam One, which is affiliated with Quest Diagnostics, if you're familiar with them. And they can send phlebotomists to people's houses, so we were sending a phlebotomist who was then mailing us blood samples so that we could keep doing our study.
Mm-hmm. Yeah, I think that little excerpt of the story really highlights the operational component of space medicine, where you have to be deployable, you have to be ready, your equipment has to be redundant, you just have to be ready for all sorts of contingencies, and it sounds like you were able to make that time point work and get some good data out of it. So that's a great little excerpt from the practicalities of, you want to study astronauts, okay, sounds great, but here's the actual reality that you're getting samples at different time points. People are busy, they're constrained, and you can only sample maybe when your patient's back on Earth a lot of the time with some of the certain tissues and the certain types of things you want to do.
That's some of it. Yeah, for sure. We want to do before flight and after flight because we need to collect the baseline before they go up. And then after flight, we want to see how they're recovering. But that getting data in flight is something that's been extremely difficult. And for the Inspiration 4 mission, we didn't have the sorts of resources that were available on the International Space Station for scientific studies. Everything that we did had to be stable at room temperature. So instead of doing venous blood draw, we were using Whatman cards to collect dried blood spot cards. For skin and body swab, we were trying to collect microbiome profiles. For that, we have a nucleic acid preservative that can keep samples at room temperature for about a month. You should refrigerate it if you want to do it longer than that, but that got us through the 3-day mission. So there's some research tools, preservatives we have available to us that can help us.
Mm-hmm. And was that RNA? Because the RNA is particularly fickle at room temperature, right?
RNA is
being degraded, and all those RNAs is running around in the air. So
it's true. Yeah. So I think we probably could. We haven't done this, but in theory, you could get RNA off of the dried blood spot cards if that stabilizes it. We don't have RNA-seq data in flight from Inspiration 4. Mm-hmm. We did get that from Axiom 2.
Oh, cool.
So Axiom 2, they had the scientific resources of the International Space Station. Mm-hmm. So Axiom Space is a private company. They're working on building their own space station. But for now, while they're working on their own station, they're sending crews to the International Space Station. So the Axiom 2 mission was a crew member. It was, again, a crew of 4 people. 2 of them were American astronauts. One was John Schaffner, and then the other was Peggy Whitson, who is a retired NASA astronaut, is now leading missions for Axiom. She's the one. All of Axiom's flights have at least one experienced astronaut flying with them. So she is like one of, if not the most experienced American astronaut.
Yeah, I think she definitely gets either the gold or silver medal on that. I think probably the gold. She's done more spacewalks. She's done, she's been on multiple different vehicles. I haven't met her in person, but she seems incredible.
Yeah, I believe she still holds the record for the American with the most number of days in space.
That's incredible. Yeah, that's so cool.
And then the other 2 crew members were from Saudi Arabia. They were Saudi Arabia's first 2 astronauts in the history of the country. So they were up there as well, also participating in this research. How we got RNA data from them was, we didn't have the astronaut time to do all the blood processing in orbit, because Axiom's business model is very similar to how you would run the International Space Station, except the ISS is funded by taxpayer money. So every time you want an astronaut on the ISS to do something, there's a certain amount of tax dollars that it takes per hour for them to do things. So their time is very expensive. And the same is true with Axiom's astronauts. You can do more with them in your scientific research, but you're going to be paying for that time. What we were able to do on that mission, we drew cell processing tubes, CPTs, from the crew, so we did get venous blood draws. And then instead of doing any processing in orbit, what they did was they just spun down those tubes in the centrifuge and put them in the freezer. And we found when we got those tubes back on Earth, when we thawed them, we could recover some of the PBMCs for single cell RNA sequencing. So doing a 10x genomics protocol. Interestingly, the scientists we were working with said, these cells look really small compared to the normal cells we work with. So I suspect there was, when cells freeze, we didn't have any DMSO in there. There was no cryopreservative.
Oh, you just flash froze them, you just threw them right
throw them in the
out.
freezer.
Yeah.
Yeah.
All right.
We didn't have the astronaut time to do
Yeah.
that with them. So I suspect since the larger cells hold more water, as the water was expanding when they were freezing, it ruptured those cells. So preferentially the smaller cells were more
Remained,
likely.
yeah. Well, that sounds plausible to me. So but yeah, that's just the little things that you learn while doing the experiment. You can, I think the saying is like, planning is futile, but planning is also necessary. Or another saying, it's like planning never survives contact with the enemy. So you do all this planning, and you can't not plan. And but all these unexpected things kind of come out of it.
The quote from, I can't remember which U.S. founding father it was, but it was like, more plan, more luck I seem to have.
Yeah, that's funny. So do you have any particular pieces of data or interesting stories you have from any one of these missions that a little inside look at what it looks like to be a genomics guru that's also working with astronauts? Any wow, I can't believe I'm sampling this in the back of a truck, or anything.
It's funny you mentioned the back of a truck. Yeah, so I mentioned we're 8 hours away from the Blue Origin launch site here at the University of Austin. It's an 8-hour drive into West Texas. There's not much really along the way. You pass, Texas wine country, Fredericksburg, about an hour from here, and then it's just desert, basically. So I went out there. We were collecting samples last summer from a Blue Origin flight. We had 2 astronauts on there that were interested in participating in SOMA, the Space Omics and Medical Atlas. That's what we've called this data compendium of commercial astronaut data that we've been collecting from SpaceX crews, from the Axiom crews, from the now Blue Origin crews. The issue, is in West Texas there aren't really any scientific facilities for us to use, and Blue Origin doesn't really have a research base built up at this point. And they're very limited with who's even allowed on site. So I couldn't even get on site for the Blue Origin flight. They, Blue Origin employees brought me blood samples and urine samples at the gate.
Oh, wow.
So I had to stay in the closest city to the launch site, which was a small little town called Van Horn. This is a city, it's very small, and it's unlikely to expand unless somebody can get more water to it. The water rights are kind of a limiting factor on how fast this place can grow. So it's a town pretty much all one-story buildings, no hotels really, only motels. So I had lab equipment in my car to process these samples with. In the trunk of my car, it was a laminar flow hood so that I could filter air coming into the workspace to make sure no microbial contamination was being introduced. And another seat of my car, I had a centrifuge to spin down blood tubes.
Oh my God.
I had freezers with me that I could plug into the ports on the car. So I had one freezer that could go down to -80°C. You have to plug it into the wall to get it down to -80, but then it can stay at -80 in the car. And I had another one for -20. And so what I was doing too with all of this equipment, there was no lab for me to set up in. So I parked outside of a motel. I ran extension cords from my car into the motel room to power all of the hardware that I had with me, and just sat in the back of my car, aliquoting blood. The owner of the motel actually came over at one point.
What the heck
to
are you
check
doing?
Because I was in the lab coat.
It's a couple years after COVID, so it's that's Wow, you're just, yeah. So what did they say?
He just looked around, he looked in the trunk, and he was like, oh, it's just blood. And then he walked off.
Okay.
So I don't know what he was expecting, but I just kept doing my thing. Nobody else came up to me, except in the evening. There's a lot of stray cats around Van Horn, so I had this little kitten come up to me and sit next to me while I was doing more aliquoting of samples. And I was terrified the kitten was going to jump into the laminar fluid.
Mm-hmm.
But fortunately, she stayed on the ground. She was actually very sweet and affectionate. I tried
Why am
to
I
find
getting all
her.
this feline DNA in my sample?
I noted it in the metadata. No, I mean I did, but there was no cat contamination. I actually tried to find that same kitten before I left and went back to Austin. I called someone at the university and I was like, "There's this kitten. Do you want this kitten?" I convinced somebody to adopt this kitten, but I couldn't find her.
Mm-hmm.
So unfortunately, she is still. Hopefully, she's still wandering.
Yeah, maybe next time
Van
you'll
Horn.
you'll have a reunion when you go for the next mission that you support. Wow, that's just the little things. Aliquoting samples in the back of in the trunk of your car, using all this portable equipment. It's like you were practicing for being an astronaut on Mars. Like
Yeah.
you're just in the back of a rover.
I don't know what the analog for a cat would be coming up to you on Mars. That
Yeah.
would be interesting. But
Yeah. Wow, that's an incredible story. Yeah, the little things that you learn while doing what you love, and takes you in interesting places. So, thank you for that. I kind of wanted to chat about a little bit more about what you're doing now, in terms of some of your active campaigns online, and how we first connected at ASMA a couple years ago, but I've been following your work on the Twitter or X sphere. Can we talk a little bit about what you're up to these days?
Absolutely. So I mentioned earlier, one of the most frustrating things about astronaut omics data is how inaccessible it is and how hard to distribute it is. So the NASA Twin study, when I first joined Chris Mason's lab, I was going to conferences and I had people come up to me and ask me where the data was. And that was really confusing to me because if you publish in science, all of your data is supposed to be open to the community.
Or readily available
Readily
by
available
request.
by request. Exactly. And so from talking to all these people, I found out it wasn't like that. And so that made me really determined for the Inspiration 4 work that we were doing to make sure that scientists could actually access it. And so all of the data that was generated from that mission, NASA has that database that the ISS data was going into that I mentioned before. So we have mouse data, sea alligators, Drosophila, microbial data. So, but they didn't have any human data. And it seemed like that was the case just because there weren't really humans that they were doing it on. So I worked with the team there to get the Inspiration 4 data into that repository, to disseminate it to researchers. And we were doing that for about half a year. All of the researchers who wanted access to it had their own institutional review boards, review their requests at their home institutions. So I was on the review panel for all of the requests coming through. So I could see all of these IRB documents coming in from the individuals who wanted to access the data. The Inspiration 4 crew themselves also were under IRB. They had gone through their informed consent process as overseen by Cornell. So we had all of this squared away. There was this pretty rigorous approval process in place. And then about half a year ago, NASA took the data out of that repository. And they told us the reasoning was for IRB reasons that it needed to go through NASA's IRB, and that it would be online once this process was complete. It's been half a year, the data is not back online. We have no update when it will be back online. So I'm currently working with my nonprofit, bioastronautics, and with Cornell to get this data accessible again through other means. Because when we were publishing it with Nature as well, we have to make that data available. We actually had to convince Nature that OSDR was a reputable data repository.
Okay, I didn't expect that, to hear that.
It wasn't on their list of approved repositories, like the NIH databases were.
So how did you go about approving that? Like,
I believe that was handled on the back end by some folks at OSDR.
okay. Hey, we're real, hey.
Exactly, yeah, they had to go back and forth and just prove they were legitimate, prove they were
Data
going to
security,
be around
yeah,
for a
yeah.
while.
Okay.
That's what they're worried about is continuity. That
Hmm.
people can be able to continue to access the data. And OSDR is actually, I think it's better than some of the other databases out there in that they're really rigorous with metadata annotation.
Mm-hmm.
So you have a really well-written description of what the samples have been through and that history of how it was processed. And I think they've put a lot of work into that because in space experiments, there's so many different pieces of this chain of getting a sample from the astronaut or from the space station and back to the researcher. So you want that, you need that extra annotation.
Mm-hmm.
But about 6 months ago, NASA took the data offline, and we've been having trouble getting that data, making it available to people. It's still a priority for me, it's a priority for Chris Mason and for other researchers in our network to make this available again. What I'm doing right now, though, is I want to start creating a pathway for astronauts who want to make their data publicly accessible. Because if you're an astronaut and you participate in this study and you expect your data to be used for scientific research, and then you're getting censorship from the government on that data, then that's not what you signed up for. You signed up for your data to be used to help advance spaceflight.
Mm-hmm. So
is there a pathway we can create where nobody can come in and censor that? And so I've been speaking with an organization called the Personal Genome Project, PGP. And this is it's a study run out of Harvard University where you get your genome sequenced and you can get other transcriptomics, epigenomics, other different types of omics related studies done on you. And as that data is posted online, your name is right next to it.
Can't obscure that.
Can't obscure that. So obviously not every single astronaut is going to want to participate in this, and I wouldn't expect any NASA astronauts to want to participate in this. But we have this emerging commercial sector where you have people who are really motivated to make their data accessible, really motivated to help lift the community, help get space exploration going, not just from all these advancements we've made on the engineering and the mechanical side, but also worrying about how the human body is going to sustain itself on the moon and Mars. And we do need a lot more data in those areas. I'm not saying that, Mars colonization can't happen until we do that, but if you want to live on Mars and you have a normal lifespan that you have on Earth, then it does.
Mm-hmm.
So what I'm doing, I've currently, I figure lead by example. That's what George Church did when he started PGP. He put his own genome online. Craig Venter did the same. James Watson did the same through their own initiatives. So there's currently competition going on with an organization called the Space Exploration and Research Agency. They're working on filling a Blue Origin field. They have all 6 seats booked, and they're filling different seats based on different regions of the world. So I'm competing for the area. It's a little unclear to me what the borders of this area are, if it's an international seat or if it's a North American seat. But I'm competing for that, let's call it an international seat.
Okay, yeah.
So I'm competing for that international seat with the promise of I will collect my genomic data. I'll do the baseline time points before, and I'll do a time point immediately on landing and put that into PGP to be this first publicly accessible open astronaut genome. Hopefully encourage others to do the same, and kick off, having a repository of data that can't be taken offline by either an academic organization or the government or whoever is holding these levers.
Yeah. I mean, what an incredible combination that you're trying to put together your personal passion for all this omics work and all this incredibly highly scientifically relevant research that you've done over your career, and then putting your words into practice on your own self and saying, hey, like I want to be a part of this this mission. I want to be a champion for open science, which is kind of an interesting scientific discussion around how do we do open science and leading the charge with your own DNA and your own being the person in that potential seat. That is so exciting. That is so. How is the journey going to continue for the next days, weeks, and months? And when does it kind of conclude? And what's the whole timeline of that?
Yeah, so there's 4 phases of the competition. Right now is phase 0. It's kind of the beta testing mode where they're still smoothing out some kinks in the process before they launch it to the wider community. So that'll be done, but that'll be done very soon, within a few weeks. So this phase 0 is basically, in phase 1, is this period where everyone who wants to go to space is spreading their message as far and wide as they can. So they're posting on social media platforms why they want to go to space and building up support, they're building up their message. And so those are connected to so it's run through a Telegram app, which I had never used Telegram before this competition, so this was new to me. So I downloaded Telegram, I opened up this Telegram app, and immediately when I opened it up, there's this leaderboard there. And everybody's name is connected to a number of points that they're calling Stardust. And Stardust gets populated based on social media views. And so you connect your accounts, you can connect your X account, connect your YouTube account. They're working on Instagram and TikTok as well. And then every single view that you get on those platforms talking about the mission will populate your Stardust, help you move up the leaderboard, and help you make it to that next phase. Phase 3 and phase 4 are when people start selecting their crew members. So what you'll do in a later phase is find the other 5 people you want to go to space with and form a team.
Okay, so if you were to be selected for that international seed, then you would choose the other 5. Is that what you're saying?
I would choose the other 5 / they would choose me. A bit of a mutual relation there. But yeah, we'd come together. You'd kind of figure out who you want to team up with, and then it'll be down selected to 3 teams. Mm-hmm. Those 3 teams are going to compete on a YouTube series.
Okay.
I have no idea what that competition entails.
I'm sure it'll change over time.
But then the winner of that YouTube series is the crew that gets to fly.
Okay, interesting. So it's like kind of a multi-phase thing, and okay, that's And when does this all kind of come to a head? When's the next big checkpoint for you?
Yeah. So the next big checkpoint for this, I mean, right now it's just making sure I get my campaign message out there, so that I can get to the next round. I would expect that next round to occur within 1 to 2 months.
Okay. And is anyone else gonna have this kind of carrot that is anyone else gonna publish their DNA open source?
If I don't make it onto that flight, I hope I inspire someone else to do this. One of the things that's just very frustrating to me is I also do a lot of teaching and education at the undergraduate level. And so this data that we have, the Inspiration 4 data, the SOMA data, you need to be a researcher under an IRB protocol to access it. So if I want to use astronaut data in the classroom, there's no way for me to do that. If we make astronaut data accessible from PGP, that changes. We can develop course curricula around astronaut data and actually start bringing education to the undergraduate level. Mm-hmm.
Yeah, removing that last barrier. What an incredible way to essentially just embody everything that you really care about all under one roof. So if you love space medicine, vote for Dr. Overbey on her campaign and watch all of her content on X and where else?
I'm on X, I'm on YouTube, I'm on Instagram, and then I've made a TikTok account, but I haven't yet posted anything, but that should be coming shortly.
Okay. Yeah. So it's incredible campaign. We'll see how it goes over time. Seems like you have a lot of exciting things in the works. You mentioned a little bit about teaching at the undergraduate level, so do you care to talk about that a little bit more?
Yes. So I'm at, as we've mentioned this, the University of Austin. It's an undergraduate institution that just recently launched. So we don't have any graduate programs yet. When I'm working with graduate students, I'm usually collaborating back at Cornell or at other institutions. So I'm working with a pool of undergraduate students here, and a lot of what I'm doing is developing coursework for this new university, it's a really good time actually to develop new coursework, because AI has kind of just totally broken the educational system across the US.
When I look at that landscape, I'm like, all right, how do you even teach undergrad anymore? It's kind of an existential kind of question that I've come across, and I'd be really curious to hear your thoughts on how you're navigating that.
Yeah, so all of our courses have, it's a good time to design a new course from the ground up, since all the other courses are break-in in some capacity. And at other universities, just because of entrenched bureaucracy or whatever the reason, it can be hard to steer these giant ships in different directions. So we don't have to break down old bureaucracy or old patterns. We can just start building and trying new things and experimenting and seeing what works. One thing that we've done here at the university is we make sure most of the assessment is done in class, where we can see them, we can see they're not using AI or any sort of assistance there. So it's designing projects where we can actually still, projects or tests where we can still see what the students are doing. One thing we experimented with last year were oral examinations.
Okay.
So
yeah.
a lot of these classes, the class I taught last year was a foundational physics class where I would give them the Feynman lectures, which are like these very dense physics lectures. They're written to be more introductory, but in practice they're still a little bit advanced. And I had key questions I wanted them to be able to answer about how the world works, like why does a satellite stay in the air? So they have to talk about orbital mechanics. Or why doesn't a bird get electrocuted standing on an electrical wire? It's because the bird's not grounded. So they would have to go through the Feynman lectures and pull out the physics principles to answer these questions, and then come in for a final exam and be able to tell me. And I'd encourage them to, and I had a structure for them to follow about how to answer these types of questions. So they at least had like a speaking format, but they had to find the physics principles and plug that in and be able to tell a cohesive answer. And they were also allowed, I encouraged them to use AI to help them break down the Feynman lectures. I said that was a bit of a dense reading. So if you have something like ChatGPT with a Claude next to you, as you're reading through those and you encounter something you don't understand, you should just be copying and pasting that over to an LLM and have it go back and forth with it until you understand. So that was one thing we experimented with. For this, I've seen the liberal arts professors doing something a little bit similar where if they have a student write a paper, what they'll do is the student will have to come in and walk through the arguments in the paper. So if they didn't write the paper, you can tell when they come in and try to tell you.
Because they're
what's
a
in the
little
paper.
flustered, and yeah, it sounds like this type of examination is a lot more effort, but instead of A, B, C, or D, yes or no, you're having to do this whole oral examination, but the quality seems like you're getting a lot of return on the effort that you're putting in, getting a lot more quality out of it. Yeah, so tell me about some of the courses that you're teaching.
Yeah, so I mentioned the foundational physics course I taught last year. This upcoming year I'm teaching, tomorrow actually is our first day of class, I'm teaching a foundational biology and biotech course. What we're doing in that course is we're starting with the big and emerging technologies that are rooted in biology and biotech principles. So we're looking at embryo selection, so like what the company Parasite and Nucleus are doing. So that gives us a chance to drill down and talk about polygenic reservoirs.
Is that like pre-implantation genetic diagnosis? Is that like
So what those companies are doing is when you're doing IVF and you generate a bunch of different embryos, how do you know the best embryo to implant?
Yeah.
So you can actually take a single cell out off of an embryo and do full genome sequencing on it without harming the embryo. So you can do that for each of those embryos, and then you can look at their how likely it is based on their genetics for them to develop certain diseases within their lifetime. So you can look at Alzheimer's risk, you can look at Parkinson's risk, you can look at more complicated diseases like autism and cancer risk, and you can pick the embryo that has the least likelihood of developing these sorts of diseases. And some of these companies, they could easily go beyond just diseases as well. I believe it's Nucleus that has a picture on their website that shows predictions for eye color. Predictions, for these more cosmetic things as well. So we're going to look at the actual data type that's being fed into these algorithms. So this might be something that students have to either have an informed opinion about to use in their own life, or maybe this is going to be something that's on the political ballot one day, and they should be scientifically literate enough to know what they're voting for or against.
Yeah, talk about a topical conversation and an interesting ethical underpinning to all that, and kind of combining science, philosophy, and kind of the cutting edge technology. So kind of seems to be a really interesting course. So I'm sure you'll have a good time this semester teaching that.
Thank you. Yeah, I'm looking forward to it. We're also going over de-extinction, so bringing back the woolly mammoth. I don't know if you've seen the little woolly mice.
I don't think so. Yeah, I don't think I have.
Oh, okay. So there's a company in Dallas, Texas called Colossal. Their goal is, it was one of these George Church companies, though it includes a bunch of scientists from all over the world now. What they're doing is bringing back extinct species. This is sort of the future of conservation, where you can take a species, maybe it went extinct recently, maybe it went extinct far in the past, and you can start to resurrect its genomics by isolating the pieces of the genome that are unique to that organism and putting it into a more modern species. And doing that enough times over time, you can get the modern species to resemble more of that historical species.
Mm-hmm.
And this is useful, for example, if you want something like a woolly mammoth again, if you want that type of organism occupying that role in, snowy tundra, you can't just put an elephant up there. You have to make it more like the mammoth.
Mm-hmm.
So what Colossal did recently, and this development trajectory to bring back the woolly mammoth, was they had isolated two different components of the woolly mammoth. One was the type of fur that they have. They have this thick brown fur. The other was a metabolism that's more conducive for the cold environment of the tundra.
So like brown fat or something that keeps them warm, or like what's,
something like that. I don't remember the details of exactly how it's working, but those were the two phenotypes they were going
Mm-hmm.
for. And they modified, I think, 6 different regions of the genome to achieve this. And they did this in a mess.
Yeah, got to start somewhere.
Start somewhere. So you can see these pictures online of these small, they're calling them woolly mice, where they have the fur of
Oh my
a woolly
God.
mammoth.
That's amazing. Wow, yeah. That's, yeah, what an interesting way to use that technology, and who knows where it'll go in the future. And it sounds like you're trying to prepare your students for that future and to keep them informed. So, more power to you for doing that heavy lifting and educating our young folks. So it's amazing work that you're doing. I kind of wanted to shift a little bit to maybe a broader topic. How would you kind of advise people to get into space medicine as a career? It's such an organic pathway. People are often very, you can't really sometimes get a degree in it, or maybe that's not the most useful thing. So how did, can you draw upon some of your experiences and just talk about how to get involved and maybe some opportunities that you know of that would be good, maybe working with OSDR or something like that would be.
Yeah, I mean, my best advice, depends where you are at which point in your career. If you're a high school student, undergraduate student, my advice to you is to become really good at some specialty outside of spaceflight. And then later in your career, or maybe simultaneously, figure out how to translate that for spaceflight. So I think what you'll find is spaceflight is a field. If you want a really rigorous genomics education, the place to start is not by looking for genomics resources from NASA, it's by going to the top genomics schools, learning everything you can about the human genome, and then piecing together how space is different. And I think that's true across a variety of different fields. If you want to understand muscle loss or bone wasting, become an expert on everything we know about that on Earth.
Yeah.
Because the programs that are out there for that are much more advanced.
Yeah, and I didn't pay Dr. Overbey to say that. I've been saying this for for many years at this point. It feels like going from an outside-in approach that you kind of just described, becoming that expert in something that isn't space flight, having to bring skills into organizations like NASA or SpaceX. They're looking for biologically minded people, medically inclined folks, and bringing those skills in, you're not going to get them from inside. They're not there yet. Those pipelines don't exist, so, or at least a lot of them don't exist. So finding those skills that you can really translate in those different fields is just incredibly important. So you highlighted my time frame or my logical way of thinking about how to get involved, and you mentioned bone and muscle loss, like I swear I didn't pay her. I swear.
Yeah, I mean, this was how I got my entry into this field. Was I was at a genomics department. I was studying cutting edge methodology for the human genome, and then I saw space flight. I saw Kate Rubins doing genomics work in space, and then I realized, oh, like this is a really ripe area to apply my skills because they're so far behind where terrestrial medicine is. So my advice, you know, once you're developing these skill sets, is then to find the hooks that you can use to get into the community. So I was working with the animal AWG, run by GeneLab, now OSDR. That was my first entry. That led to an internship at NASA, which led to me knowing about the postdoctoral fellowship application, which led to Chris Mason's lab, which led to, you know, the inspiration for a nature package.
Yeah.
So there's that as an opportunity. Check out the AWGs from OSDR. The other thing was, through my nonprofit BioAstro, we run a summer research program that we call a crew program. So we recruit a cohort of people every year. It's pretty competitive. We last year had about a 10% acceptance rate into the program. What we do is we curate a collection of PI's projects, and then we look at the applications that we get. We look at the skills people have. So again, what you're bringing to the table outside of space knowledge. We pair people with skills to the principal investigators that have projects within that skill set.
Yeah, just kind of matching skill set to project, and I'm sure it's all one-to-one and it's super easy, and everyone's has all of the skills related to all the projects that come in. I am joking, of course. I'm sure that's a difficult program to put on. But any like highlights from that, or any stories that have come from that? Because we're recording here in the middle or early part of September. So did that just recently conclude or anything like that?
Yeah, it finished up. We concluded last week. We had 10 researchers in the program over the summer. All of them are continuing to work on their projects even after the fellowships ended with the PIs that they were matched with. So I mean, that's a huge success from our point of view, that, you know, the PIs want to keep working with the researchers keep wanting to work with the PIs. I was very clear to them, like, you are not under any obligation to keep working. You can diplomatically leave, like, that's totally fine.
Yeah, yeah.
And they all kept working anyway, so that's so inspiring and encouraging to see. I hope that we make a lot of long-term connections through the program, and I'm really hoping for it to serve as an entry point, and this is also as is encouraging for people who haven't done a lot of work in space research to get a hook into it and give themselves momentum to keep working. So seeing them continuing to work on the projects shows me that that's working. Some of the projects that they're working on, for example, BioAstra has a program we're developing called Orbit, which is working on ways to rapidly deliver different types of medical supplies in different situations. So in a humanitarian crisis, or in a urgent military situation, whether or not we do this terrestrially, or whether or not we can create a constellation of medical equipment orbiting Earth.
So cool. what a sentence. Yeah, that's amazing.
And so one of the really great connections we were able to make was that we had people apply to the program who are working at the Air Force's medical division. And so being able to take people who have been working on the science side with the Air Force and being able to help them have eyes on the Orbit program, and then also the Orbit program giving them structure for how to think about these challenges and connecting them with the people that you need to talk to, ended up being a really, what's the opposite of a parasitic relationship? Where both people are getting something in return.
Mutualism? Or, yeah, I don't know.
Yeah. Both people are, like, both BioAstra and I think both the researchers from the Air Force division were able to benefit from this.
Yeah, I think that story really highlights how important it is to both have data, but also mentorship to understand how to structure the data, what the other side is going to want out of a given project. You know, you can come up with a X, Y, or Z research projects, but making sure that it fits in with the stream of what someone wants down the road or what's even topical is so important. So Finding mentors through the crew program seems like a great option, and just seeing people online, finding people out there that are in the community online, probably looking for help on all their research projects. It's really important.
The best way to follow the crew program too, bioastronaut LinkedIn is our primary mechanism where we deliver updates to the community. So if you follow bioastro's LinkedIn page, you won't miss the solicitation for our call for Crew 4.
Yeah, very cool. Yeah, there's usually a big, whenever I talk to non-space medicine folks, they see a big crevice or canyon between, they're interested in the field. They're like, wow, what you're doing is really cool. You're looking at bone loss in space, but how do I get involved? What does that even look like? Even if someone has an online tool, so trying to get that gap filled through all these programs that you're mentioning and working on and have recently completed is just amazing. So just hats off to you for all the work that you're doing. It's amazing. So moving on a little bit more, I wanted to talk a little bit more about some of your recent research findings, anything that you find interesting, like any highlights of like a recent paper or like a recent finding, like, oh, this was upregulated and we didn't expect that, or any paper that you've published recently and your favorite kind of highlight.
Yeah, I mean, my favorite highlight, particularly, I mean, so much work was published last year for Inspiration 4.
There were so many nature papers.
There was so much.
I was like, repost, repost, repost.
We had a lot come out. My favorite finding from that package in particular was from the skin biopsies that we did on the Inspiration 4 crew. So this isn't a data modality we've gotten from any other crew member, crew from a different flight so far, but we got it from Inspiration 4. And what we were really keen on looking at there was markers for inflammation. What we saw in the NASA Twin study, when Scott Kelly came back to Earth, and he wrote about this in his book Endurance. I think he also wrote a research paper with a couple of people talking about his symptoms. He had a really bad skin reaction.
On his feet, right?
All over his body.
Yeah.
He would report that just the clothes on his skin would create a burning sensation. So we didn't, have any omics profiling of Scott Kelly's skin from that mission. We had these skin biopsies from the Inspiration 4 crew, and what we did with those was we worked with a company called NanoString. They make spatially resolved transcriptomic profiling tools. So what this means is that you can actually see gene expression at spatial resolution. So if you're doing something like 10x genomics and you're using a microfluidic system, you have to dissociate the cells, you lose their spatial configuration relative to one another. NanoString, what you do is you'll slice the tissue, you'll get it on a slide. You can, using a computer program, draw the area around that you want to sequence. So I was manually drawing different layers of skin, because the skin, when you put it on a slide, has a very kind of irregular structure. So I was figuring out, we had different markers that we used to stain different layers, so I could see this is the epidermis, this is the dermal layer, this is the vasculature. And I separated the outer epidermis from the inner epidermis when you're getting more cellular regeneration. And so we looked at gene expression across different layers of the skin tissue. And we could see, even though none of the Inspiration 4 crew reported any sort of inflammation when they got back, we could see inflammatory gene expression signals in the different layers of skin.
So like, leukin 1, leukin 6, like all these inflammatory cytokine, like circulating proteins you're talking about, right? Yeah. That's if you have ever seen spatial omics work, just do a quick Google search. It feels like you're looking into the future where you're actually able to see the tissue level and also the expression on top of it. I can't really explain it better than that at this point, but it really feels like the future because it really gives you that the marriage of structure and function that biologists maybe talk about in classes, but actually bringing that home visually is so important for research and as well as application.
Yeah, we're applying some of that same technique to eye tissue that we've gotten from mice from the International Space Station, so we can look at the different regions of the eye spatially as well. It's not published yet. I'm still analyzing it yet, so I don't have any results to share. But I think that is the future of, especially when you can pull apart something like a mouse and actually see every organ, the way that we're obviously doing with astronauts.
Yeah.
It becomes a really powerful, I think, next generation tool for space research.
Yeah. Yeah, I guess I quickly wanted to back up and talk about the OSDR, which is the Open Science Data Repository. So by the nature of the name, you may intuit that it is open source. People are wondering where do I find data on how to look at the effects of unloading or radiation on your favorite tissue of choice. I think free of charge you can sign up and get a boatload of data that you can analyze. It's there.
Yeah, you don't really need to sign up we had mechanisms put in place for accessing the Inspiration 4 data because that was human data, but for all the animal data, for all the fruit fly, worm, bacteria, whatever else you want to get from there, you don't even have to make an account. It's just an open repository. You can start downloading data immediately.
It's just so important to get like almost every space medicine paper I've read, I always talk about like the small n problem, and it's been this kind of classic paradigm with space medicine research where you have a crew of the shuttle up for a week or something like that. There's 4 samples from an astronaut, and they have a hard time extrapolating for larger subsets, or if you have bigger questions to answer, you usually need more people to power your study. And so really digging into that depth, the future is bright in terms of increasing that N and including your own DNA hopefully in the future. So I think as we go forward into the future, we'll be having more and more data to look at. So really exciting time. I wanted to also talk a little bit about like your perspective on like NASA, SpaceX, Blue Origin, your role in the near future. What are your big views on getting us to the moon and Mars? Just kind of like the bigger questions. Like we've talked a lot about your work, your journey. We've talked a little bit about some of the activities that you're doing now. But like, what's kind of your sci-fi future that you see us going to in our careers as we become more and more involved in space medicine?
Yeah, my opinion so I do a lot of work on astronaut health. I want to make astronauts as healthy as possible for their missions. And I think we have a lot of basic science research we need to do on the genomic level in order to actually design drugs and therapeutics for astronauts. But I don't want people to take away the message that I'm opposing any sort of barrier to spaceflight, like saying we can't fly until we figure this out. I think if you're thinking about a moon mission or a Mars mission and you're a private citizen and you know the risks and you want to do it, you should do that, and the medical equipment that we send with you should be focused probably primarily on emergency care. Something comes up, you need to fix it. Here's what you need to do to fix it. Or how do we mitigate or manage, reduce the possibility of disastrous outcomes happening? Like, do you need to get your appendix removed before you fly, for example? So I think the work that I'm doing isn't meant to be a barrier to say that we shouldn't go to Mars until we figure this out. It's helping us get to the point where we can live healthy, normal lifespans on Mars, where we can get to the point where we can reproduce on Mars. The sort of things that you want when you want to have a healthy, self-sustaining, multi-generational settlement.
Yeah, I think that's a really important thing to reiterate, that it has come up in many different contexts as I've explored this area of medicine. Most particularly with the FAA, they're trying to change the conversation for medical optimization to get more people to fly and be pilots, rather than people that are taking away people's wings to be able to fly. And so I think if you were to talk to many space medicine researchers, I think they would totally agree with you that they're not trying to hold people back, but to get more people to do it in the future. So it's a way to accelerate space travel and make sure that we can do it because of all of these negative risks that happen when you get into that isolated, confined, hazardous environment of unloading and radiation. Humans weren't built for that, and we have a lot of figuring out to do before we can do it in a way that is going to be sustainable, and so yeah, like talking about that generational kind of ideal that's brought about in like the SpaceX community. We're going to have this generation living and working on Mars. A lot of talk is about the engineering, and a lot of the passion that I see from most of the people I've talked to is that the biology needs to, like, we need to engineer that biology to catch up and to be as strong and as to be as fit to live on in those places. So we have a lot of work to do, and we really need more and more biologists, physicians, all sorts of healthcare folks to get involved in ways that we won't even know how exactly to do that in the near future, in terms of there's going to be jobs in the next 5 or 10 years that we could never have anticipated. So just joining the discussion, getting involved in research projects is going to be an incredible journey. I really feel like there's going to be an arc in the next like 15, 20 years where space medicine is going to kind of take off. Obviously super biased, but I think it's really, the enthusiasm is building as we have that engineering component that is getting figured out about 4 hours or 5 hours south of here. Having Starship, having all of this space. In my talk with some researchers, they're like, imagine all the science we could put on that thing. 100 tons to low Earth orbit, like it's like a dream come true. We've been sitting working in a very tight box, and we've had to be ingenious about how we use that box and how we study those folks in there. But as we engineer a larger and larger box, it's just going to be a very bright future. So
yeah, I agree with that completely. I mean, if you have dream experiments you've been wanting to run, I mean, figure out what you're going to need. Just start planning for them now, because it's gonna be much cheaper to get things to low Earth orbit very soon. I mean, the other aspect is too, and this is maybe where I differ from some people at NASA, is I think we're not going to have perfect resolution on which biological problems we should be tackling until we start getting boots on the ground in various places. I think sometimes researchers want to figure out the problem before we send the person, especially NASA as an organization is very risk averse. Whereas I don't see a huge ethical issue if somebody is aware of generally the risks of spaceflight and they want to go to these destinations anyway, that we shouldn't be stopping them from doing that, but we should be making sure that we can study whatever goes wrong to make sure it doesn't happen for the next person.
Mm-hmm. Yeah, I think that brings up kind of like a structural component of this landscape that's shifting between a civil servant and a private astronaut, and kind of the ethical underpinning between what are their obligations to risk under those professional roles. So I think that as commercial space flight becomes more and more routine, a lot of those barriers are going to be kind of coming down, and we're going to have to figure out how to do it all safely and how to navigate all of this. And people that are interested in philosophy, we're going to need you too to help us figure out all these ethical considerations. So yeah, that's it's an incredible time to. I think I really see a growing interest and a bright future in. It's like the most interesting field of medicine to start with, but also kind of tackling these interesting questions, and it's going to go through a lot of figuring out to do. And whether you're interested in kidney disease or hypertension or diabetes, they all have an interesting consideration for space flight. So we're going to need all of our different minds to come together to help people live in that beautiful future that we all hope for.
Well said.
Yeah. So Yeah, so I kind of wanted to talk a little bit more about some of your interactions at various space companies, so like NASA, SpaceX, Blue Origin. If there was another PI in the room, what is some of your advice on like approaching them or like working with them? Like, it feels like you have to have someone on the inside that you kind of can interface with, like from someone who's completely naive to a lot of that. How do you approach private space companies for research projects?
Yeah, so for the Inspiration 4 mission, I think the way that was worked out was because they were working specifically with the lab that was working on the twin study. So Inspiration 4 didn't really have the research opportunities channel figured out yet, but I know SpaceX has evolved since then. So now for FROM2 that launched earlier this year, for an upcoming collaboration between SpaceX and VAST, they actually have a request for research proposals like that they put out as a company. And what you do is you submit to them, you tell them what it is you want to do in spaceflight and why. And it's not quite as complicated as something you would submit to NASA or to the NIH. It's not like a huge grant proposal. It's more like a description where you want to show that your project has scientific merit. You want to make sure it's perfectly clear to them what the crew is being asked to do. And you want to have both enough technical detail in it to demonstrate merit, but also make it general enough that a more layperson could understand as well. Because SpaceX is an organization, at least not yet, maybe as they mature this will change, but they're not putting together panels of experts to review these. They have a team that's generally pretty scientifically literate, but isn't going to be specialized in every single proposal that's coming in.
And that's where you come in, right? You bring in that expertise in and saying, hey, I'm an expert in X, Y, Z field. I have this new technology that's, an important consideration is like power requirements, it being like operationally small, so it can fit into the Dragon capsule, which is the current architecture. And so yeah, I think everything that you just said was really important. Like having that pipeline, that RFP was, I think it was like last January or February that it got published or was brought out.
It was, yeah. I got a lot of personal messages from people during that time because they'd see me working on Inspiration 4, bouncing projects off of me. And I would look at what the things they were proposing, and I think there were two different types of people I was seeing. One was a group of people who'd done projects on the International Space Station, and they were used to doing spaceflight experiments, but they were used to having all the resources available to them that the ISS has, which SpaceX free flyer missions like FROM2, Polaris Dawn, Inspiration 4, they're in the Dragon capsule the entire time. They're not docking with the space station. So the sorts of power requirements, the sorts of like whether or not you'll even have access to refrigeration space, I was, advising them not necessarily on like, the nitty-gritty details of what's in the Dragon capsule or anything like that, but just saying very vaguely that you need to keep in mind like what sort of constraints there are in different vehicles. So what are you going to do if you don't have access to cold stowage? What do you do if you can't power that centrifuge? Getting them to think along the lines of reducing their experiment, figuring out how far they could reduce it. Like what are their actual technical requirements that are deal breakers? And then you have another group of people who are terrestrial scientists, let's call them. They've only ever done experiments on Earth. And they have no idea even how to think about microgravity as a concept. So they're like, I want to fly the cell culture dish. Like, you're not going to fly a cell culture dish. You have to have somebody have a chamber for you so that the media is not flying out. So I had to walk them through how to think about differences in fluid dynamics, differences in how operationally their experiments are between the ground and between space. This brings up another thing. We've been talking a lot about SpaceX. I've been working with a company recently. I mentioned them earlier because I'm going through their astronaut campaign channel, and I want to keep that channel separate from this other channel I've been working on with them. But they want to fly science on that mission. So they want to have scientific experiments that researchers from different countries can fly. So stepping back and going over this mission again, they've booked a Blue Origin flight, and they want to be able to send astronauts from different countries that haven't had an astronaut before. So I said I'm going for the international seat because the US has had many astronauts. But for example, they're sending Nigeria's first astronaut. When they announced the campaign, this has changed, but when they announced the campaign, they were sending India's first astronaut, and this will be India's second astronaut technically. And they're setting up regional areas, to promote having astronauts from countries that haven't gone before. So what's also happening, separate from the astronaut campaign, is that each of those seats will also be reserved for science from these different countries. And the question is, how do you design a scientific solicitation such that we know we can get valuable science out of it and people from these countries can contribute something meaningful? So I've been working with them to set up competitions where I've set up the parameters of the competition such that we know we'll get meaningful science out of it, but they can steer the direction of what we're studying. So what we're doing is we have cell culture boxes that we'll be putting under the seats of the Blue Origin flight, but they get to choose what cells go in there.
Mm-hmm.
With constraints being the constraints of that cell culture box and the constraints we receive from Blue Origin. So for example, because there's people on this flight, they're going to be BSL level 1 type cells. We're going to make sure they're constraints of the box. It needs to be adherent cells, so no suspension cells. And they need to be able to grow flat, so that takes out a lot of organ research. So flat, adherent, BSL level 1 cells. And then we look at the competition entries. This competition portal will be going live in a few weeks where people can start submitting from those countries and from all over the world for the international seat as well, where they can propose what type of cells they want to fly, why they want to fly those type of cells. And then we've set it up so that while that box is in flight, it'll release a cellular preservative. So we're going to release formaldehyde during flight, and then have it cleared away with PBS, so we'll have fixed cells coming back. And they also get to propose what immunofluorescence markers we should use to stay in the cells. So that's one way of setting up. SpaceX is a lot more open-ended, where they want all these ideas. When we're trying to expand the amount of people accessing space who perhaps haven't done a lot of scientific experiments before, it's important to set up these barriers and constraints so it becomes a little bit more narrow. The other thing we're doing, because I talked about fluid dynamics, which was something I've advised people when they're designing experiments for flight, like you have to consider how fluids are going to move.
Yeah.
Which is very important for molecular biology, because so much of molecular biology is just moving fluids around to different tubes and doing different things with those tubes. So the other competition that will be available is one that the actual astronauts themselves are going to have a part in. They're going to be executing this experiment. So the competition is to redesign the 1.5 millimeter Eppendorf tube and get it to better hold fluid in space.
Okay, yeah. Kind of like kind of reminds me of the espresso machine that was on the ISS, or the espresso cup that they had to manufacture, and some liquid would actually pour out of it. And so is it going to have like I'm just imagining a little Eppendorf that's looks like that cup.
Yeah, well, I mean, like, we still need to have the cap closed. I mean, they could redesign the cap to have it be a screw top instead of a pop top. That's if they need to change the configuration, and that would help. What we'll be doing here at the University of Austin, though, is we have a 3D printer. We're going to be 3D printing the winners of the competition, driving those, winning designs over to West Texas, giving them to the astronauts that go on that mission. Training them. What they'll do in flight is they'll have 3 tubes filled with different volumes of fluid. We'll have 1/3, 2/3, and all the way to the top. So they'll open them up sequentially. We'll have a GoPro camera watching this. And they'll go through a series of motions, moving that tube around, trying to see if and when the fluid flies out. So they can actually get visual confirmation that this tube can or cannot hold fluid, the one that won the competition.
Very cool. Yeah, well, that's that would be a fun Blue Origin webcasts. Seeing the astronauts, obviously unbuckling, looking outside, doing some tumbles, and then in the corner, some people like moving these tubes around. That'll be definitely one to watch for sure.
I think so. I think the workflow is they get up there, they're in microgravity, they're gonna stay strapped into their seats and do the tube exercises, and then put that away and then enjoy the rest of their time in microgravity.
Yeah, that's smart. All right. Well, we are approaching orbit. We like to record these orbits for about 90 minutes, which is what it takes to go around our beautiful planet Earth. So I kind of wanted to focus a little bit more towards closing thoughts and some extra things that I may have not brought up before, and kind of get us towards splashdown. So yeah, kind of what. If you were to talk to either PIs or people in research to get involved, what is kind of like one thing you would say to them? Just like a short sentence, like you can do it, or like it's not as hard as you think.
It's harder than you think.
It's harder than you think.
Well, I think it's getting easier. It's easier now than it used to be.
Okay.
I won't say it's easy, but it's easier now than it used to be. And that's because we have so many more commercial companies that you can get involved with. So before, if you wanted to do any sort of spaceflight research, NASA was your channel. Or if you were in Europe, ESA was your channel. If you had these government channels you had to go through. Now there's SpaceX, there's Blue Origin, there's Axiom, there's VAST. Apologies to all the space companies I didn't just name them in that sentence, because there's so many now. You have options.
Yeah, I think as the number of airliners come online, we'll have more and more opportunities for research, and they're going to come in all shapes and sizes, and it's going to be a really, really exciting future. And so if you're interested in doing space medicine research, there's only going to be more opportunities. And then in terms of clinical medicine, there's also going to be more opportunities to take care of astronauts, either from a preventative medicine standpoint or from more of an emergency medicine standpoint, taking care of those acute injuries or acute kind of biological changes that occur while we go to space. So yeah, I think this whole discussion is really hammered home just kind of all of the different ways that you can be involved as a research scientist, doing some teaching regarding it, trying to be a subject yourself in your own research, being an astronaut. You kind of have this incredible weaved story of how you've gotten to your particular station here at University of Austin. I just want to say thank you so much for your time. It's been really nice to reconnect with you. We first met a couple years ago at ASMA, so the Aerospace Medicine Association in New Orleans. And it's just been such a pleasure to get to know you a little bit better. Really, humbled and honored that you would want to come onto my show and just talk to me. And I'm happy to have call you a space nerd friend. And I'm looking forward to seeing what you do in the near future.
Thank you. Thank you so much for coming here, for talking to me, and thank you for visiting the University of Austin. I know we don't have any students on campus today, but any initial impressions of the university?
Yeah, so we're currently downtown Austin. It is a great city to be in to start. If you love barbecue, had some Japanese barbecue in East Austin last night. Went to go see some jazz. Was just having a really, really nice time. And then coming to campus today, it's been a really beautiful building. You walk in and it's kind of has this historic kind of feel to it. Seeing all the activity in terms of the new offices, the new opportunities for students to be in an environment that feels very focused on the student experience. Having, I went to essentially large public universities, state universities, and a private medical school, and then at different levels of education system. And this feels like you're trying to do something different here, which is kind of in the line of space medicine, trying to do something trying to tackle a problem from a different angle, and so I can really appreciate all the effort that you guys are putting in, and excited to see what you're up to in the near future.
Thank you. Come back anytime.
I'll definitely take you up on that. It's been super fun to get to know you better, and I just really want to thank you again for the gracious invite.
Yeah.
Do our little outro.
Yeah, thank you so much for coming. I never pass up an opportunity to give someone a tour of this place. I think it's really special. I just wish we had more people walking around. It's a Sunday afternoon, so
Yeah.
it's kind of a ghost town around here. But
Yeah, I mean, it's maybe better for the sound quality of this recording, so it's all good for me. And yeah, it seems like there's so much in the areas that I was able to see, just a lot of excitement about what's going to be, and the future seems bright. So thanks again.
thank you.
All right, thanks for listening to Let's Talk Space Medicine. If you want to continue the discussion and meet more fellow space nerds, be sure to check out talkspacemed.com. And thanks, and see you next orbit.